Hello, I'm Alyssa Shawn, and today I'll be talking about determining edaphic factors of Vertic clay communities. And edaphic means relating to soil. And over the past few weeks, both Skylar McKinnon, who's talking right after me, and I worked with Emma Fryer on projects related to her thesis research. And for this project, we looked at annual flowering plants of the San Joaquin Desert, which are the species that make up California's famous super blooms. And here's a picture of the super plumes. The factors we considered in this project were broken up into abiotic factors, which are non-living environmental factors, and biotic factors, which are factors caused by other organisms. The abiotic factors we looked at are the edaphic factors that I mentioned earlier, and in this case are acidicity, which refers to the levels of sodium in the soils, shrink swell potential, which refers to the change in size when the soil is wet, so when it swells, versus when it's dry, when it shrinks. And soil texture, since these species all grow in soils that are very high in clay content. The biotic factor we addressed was competition, specifically in the form of the invasive annual grass Bromus madridensis. All the species in the study are endemic to or found on Vertic clay soils. Vertic clay soils have high shrink swell potential. This makes Vertic clay very physically extreme for plant roots. In the San Joaquin Desert, where our study species are found, these soils have over 50% clay with varying acidity, and they often have high levels of selenium, sodium, and gypsum, which make it chemically harsh as well. This spring, Emma conducted 3 different greenhouse studies. My project used plant material, soils, and data from these studies. The first included these 12 Vertic clay endemic annuals of the San Joaquin Desert. And these species were each planted in 3 different clays varying in acidity, which included non-sodic, sodic, and extremely sodic clay soils to address the chemical properties of these soils and their effects on these plants. And just as a disclaimer, these are classified as soil types because we haven't gotten lab results on other specific properties. And for a second study, this is repeated with each plant grown with competition from the invasive grass species Bromus madridensis. So each of the species was grown in 3 different clays varying in acidity, and each species in each clay was grown both with and without the presence of that invasive grass. And for my project, I was looking at how soil properties and competition affected the plant's roots. For the third greenhouse study, there were 5 congener pairs that included sister species, where one of the species in the pair is endemic to our Vertic clay soils, while the other is closely related but endemic to either sand or talus. Clay has fine particles, while sand and talus have much larger particles. And each of these species were planted in the soil they are endemic to, as well as their congener's native soil. So, for example, Calanthus anceps is endemic to clay, and Calanthus laciophilus is endemic to sand. So both species were grown in both clay and sand to see the effect of soil texture. My hypothesis for this project was that plant roots physically differ in response to soil properties at these sites. And these soil properties that I described in my hypothesis include soil texture, shrink swell potential, and acidicity. I also took into account competition with Bromus madridensis. I looked into it, but it is not a soil property. And now I will go over what I did during the summer, during the Frost Fellowship. To establish the soil shrink swell potential, I conducted tests for all the soils collected at different field sites, plus the soils used for greenhouse studies, which were also collected from field sites. COL stands for coefficient of linear extension, and it measures the shrink swell potential of soils. We wetted the soil until it was saturated, and then allowed it to fully swell. We then squeezed them into uniform rods and measured the length, as you can see, and then we allowed the soil to completely dry and shrink back down, and then measured the length once more. And in the top left corner, there's the equation we used for COL, and the higher the COL, the higher the shrink swell potential. We conducted COL on the bulk soil samples used in the greenhouse studies. The non-sodic and sodic clays had very similar COLs, while the extremely sodic clay had a higher COL. Sand and talus basically have a coal of 0 because they have larger particles than clay that doesn't allow for shrink swell to occur. We also did field work in Carrizo Plain National Monument twice to collect more soil samples to tie our studies to the community. With these samples, coal will be conducted so we can see how that property relates to the species present at each site, in addition to its effects on the greenhouse study plants. And back at the lab with the greenhouse study plants, we separated them by parts: vegetation, roots, and reproductive parts, to see how different plant parts biomass differed across treatments, and to test different parts for upcoming planned lab work. And then after, I massed all the plant material to quantify the performance of these plants under the different treatments. Now I will be talking about my results. We conducted a general linear model analysis for each of these 12 Vertic clay annuals in the soil chemistry study, with soil type as an abiotic factor, which included acidity and coal, and competition as a biotic factor. We first analyzed the effect of non-sodic soils. Non-sodic soil did not have a significant effect on any root biomass for any of the study species. In addition to roots, I ended up looking at the shoot mass to see if there was a difference in plants' responses by plant part. However, as you can see, non-sodic soils only had a positive effect on monolopious strictus shoot biomass, as you can see in the bottom right. Then we went over the sodic soils effect, and for these species, sodic soils significantly affected root biomass. When looking at shoot biomass, these species were significantly affected. And for these species, extremely sodic soils significantly affected root biomass. All the species increased in root biomass when grown in extremely sodic soils. Then we observed the shoot biomass. These species were significantly affected by extremely sodic soils. I then looked at competition, or the presence of Bromus matrotensis, and for every single species, competition affected root biomass, and it had a negative effect in all the cases. I then looked if competition affected shoot biomass, and once again, every species had a significant negative effect. When accounting for both competition and soil type in our GLM, we saw there was a significant effect on these species' root biomass, which was greater than the summed effect of these factors without accounting for the other. When looking at the interaction affecting shoot biomass, it was once again very similar to root biomass. Since all three soil types in our greenhouse studies had very similar coals, it appears soil chemistry plays a large role in these communities. We will be sending out samples for lab analysis so we can continue looking into and incorporate other soil properties. I was originally looking at roots, but as we saw, root biomass and shoot biomass appear to be very similar. Overall, Edaphic factors appear to have a significant effect on these species in this community, and competition further shapes it. I also looked at the 5 congener pairs and conducted a t-test to compare roots and shoot biomass across soil types for each species. The top 5 species are clay endemic, and the bottom species are sand or talus endemic. In these species, root biomass was significantly higher in clay soils. Consequently, root biomass was smaller on sand or talus soil. When we look at shoot biomass, we saw more significant differences, but in all cases, it was higher on clay, even for sand natives. Consequently, they all had smaller shoot biomass on sand or talus soil. All these results were very surprising because the plants did better in clay regardless of the native soil. This may be due to watering methods or water retention potential. Based on our results, soil texture plays a role in plant performance, but it may be indirectly through water dynamics, and further experimentation is necessary. And lastly, a big thank you to the Frost Summer Undergraduate Research Program for this amazing opportunity. Thank you for Emma Fryer, my Frost supervisor, for allowing me to help you with your project, and so much more you don't even know. Also, thank you to Professor Nishi, my faculty supervisor, for presenting this experience to me, and so much appreciation to Skylar McKinnon, my fellow Frost undergraduate researcher, for everything. Also, a big thank you to our volunteers, Dylan Armitage, who helped us with R and statistics, Michaela Johnstone, a high school student volunteer, Elijah Balderas, who is also a grad student, and through this experience, we're also able to partake in a lichen survey for his project. And also Dee Dee Giles and Izzy Mitchell, who helped with this project. Thank you for listening, and sorry about the background noise.